Welcome to the deep dive. Today we are unpacking this, frankly, massive stack of biological research and historical documents that one of you actually provided for us.
Yeah, it's quite the pile of papers we've got today.
It really is. And our mission here is to explore the invisible, entirely passive physical forces that are keeping you alive right this very second. We're looking at diffusion and, osmosis.
Right, which sounds like high school biology, I know, but it's so much deeper than that.
Exactly. Because usually when we talk about how the human body works, there's this expectation of incredible precision, right?
Yeah.
You think of your heart pumping, your lungs expanding, and it feels like this carefully orchestrated machine.
Yeah, like every valve is opening and closing on a strict schedule. It is really comforting to think of it that way.
Totally.
We like to imagine there are these tiny biological traffic cops inside of us just pointing and saying, oxygen goes here, water goes over there, right on time.
But the sources we're looking at today just completely shatter that illusion.
Mm-hmm.
When you actually step into the world of microscopic cellular transport, those traffic cops are gone. The careful engineering just vanishes.
It really does.
We're looking at a physical landscape that is honestly just pure, unadulterated chaos.
Chaos is the perfect word for it. I mean, it really is the absolute definition of statistical inevitability, just masquerading as deliberate design.
Right.
The actual mechanics of how substances and water move down their gradients and doing it without using a single drop of cellular energy, by the way.
Which is wild.
It involves a 200-year scientific argument, a literal Nobel Prize-winning plot twist, and it's the very fundamental reason why human beings even evolved to have a heartbeat.
So let's start with the basics, because before we can understand how this works in your blood or your kidneys, we have to look at the microscopic physics of it all.
Right, we have to scale way, way down.
Yeah. So if I drop a single drop of red food coloring into a still glass of water, I don't have to stir it. Eventually, the entire glass just turns pink. That is diffusion.
Exactly. The net movement of particles from a region of high concentration, so the initial dark red drop you mentioned, to a region of low concentration out into the clear water.
Right.
And the engine driving that movement is pure randomness. It really all comes down to the second law of thermodynamics.
So we're talking about entropy, the tendency of things to just spread out and become disordered.
Yes, which happens because particles at room temperature are always in constant random Brownian motion. They are blindly bumping into each other all the time.
It's bouncing around.
Endlessly. So over time, those food coloring molecules will spread out simply because, statistically speaking, there are infinitely more ways for particles to be spread out than there are for them to remain clustered tightly together in one spot.
So no single dye molecule is ever deciding to move to a lower concentration.
No, not at all. It's a statistical guarantee based on trillions of random collisions.
And because it's just based on ambient thermal motion, like just kinetic energy, the body doesn't have to spend any ATP, any energy at all to make it happen. It's totally passive.
Completely free energy for the body, yeah.
Which brings us to the second force we're looking at, which is osmosis. Now, as I understand it, osmosis is just a special case of diffusion, right? But it's specifically for water moving across a selectively permeable membrane.
That is the standard textbook definition, yeah. Water moves across a barrier toward a higher solute concentration.
Okay, so I have to play devil's advocate for the listener here, because the language we use for osmosis is incredibly misleading.
Oh, it's terrible.
Right. When I learned this, it was always described as the salty side of the membrane sucking the water across, like the solutes were little magnets, or there was some, biological vacuum cleaner pulling the water toward the higher concentration.
Yeah, that language is actually a huge hangover from 19th century physiologists. They loved that magnetic framing. They loved the idea of a pulling force.
But modern biophysics rejects that, right?
Completely rejects it. Nothing is pulling. Nothing is sucking.
So then why does the water move? If there is no pull, what is actually happening at that microscopic membrane?
Okay, think about those blind collisions we just talked about. Imagine you've got pure water on one side of a membrane and salty water on the other side.
Okay.
On the pure side, you have nothing but water molecules bouncing against the barrier, just water hitting the wall. But on the salty side, those dissolved salt particles are physically taking up space right at the membrane surface.
Oh, so they're physically blocking the water.
Yes, and even more importantly, they are forming hydrogen bonds with some of the water molecules on that side.
So the salt is essentially distracting the water.
That's a great way to put it. It lowers the water's chemical potential. Because the salt is in the way and it's grabbing onto the water, fewer water molecules on that salty side are free to randomly collide with and pass through the membrane.
Oh, I see.
Meanwhile, the pure side is just relentlessly hammering the barrier with free water molecules. So more water randomly bumps its way in from the pure side than can bump its way out from the salty side.
It's purely an imbalance of random collisions.
Yeah.
The pure side is just throwing more water at the wall.
Exactly. It's just a numbers game.
That completely changes how I visualize this.
Yeah.
But, it also raises a massive historical question for me. If there is no magical pulling force, and it's all just invisible molecules randomly bumping into things, how did anyone figure this out before modern microscopes?
It's crazy, right?
It must have looked like literal magic to them.
It totally did, which takes us all the way back to the 1820s. There was this French physician named Henri Dutrochet, and he built one of the very first osmometers.
Okay, an osmometer. What did he use?
He took an animal bladder.
Wait, a dead animal bladder?
A dead animal bladder. He filled it with a heavy sugar solution, tied it to a glass tube, and submerged the whole thing in pure water. And without him doing anything, the fluid level inside the tube began to rise up against gravity.
Just from the pure water blindly colliding its way through the animal tissue. That's wild.
Yeah, and Dutrochet coined the terms endosmosis for fluid moving in and exosmosis for fluid moving out. And the reason this was so revolutionary is that he was actively fighting the prevailing scientific belief of his era, which was vitalism.
Oh, vitalism. The idea that biological fluids only moved because living organisms possessed some mystical vital force.
Exactly. They thought you needed a living spirit to make things happen. By using a dead animal bladder, he proved it wasn't a living spirit doing the work. It was a physical mechanism.
A purely mechanical reality. That's so cool.
It really is. The terminology we use today actually gets locked in a bit later, though, in 1854, by a Scottish chemist named Thomas Graham. He coined the word osmosis from the Greek word for push or thrust.
Which is funny, given our whole conversation just a minute ago about how it's pushing, not pulling. He actually had the right idea with the name.
He really did. Graham was a pioneer here. He was obsessed with turning chaotic chemistry into strict, predictable rules. He figured out that gases diffuse inversely to the square root of their mass.
Which is known as Graham's law, right?
Right. And he also figured out that some substances diffuse incredibly slowly, he called those colloids, while others, like salts, diffuse easily, which he called crystalloids.
Okay, so that classification is basically the foundation of why a membrane can be selectively permeable in the first place.
Precisely.
Because, if everything diffused at the exact same rate, a membrane wouldn't do anything. It would just be an open door.
It would just be a hole. But making this all mathematically predictive didn't happen until a 26-year-old German physiologist named Adolf Fick came along in 1855.
Fick's laws, right. What did he do?
Fick was brilliant. He looked at how particles spread out and noticed a parallel with how heat spreads through a solid metal bar.
Oh, interesting.
Yeah, he borrowed the mathematics of heat conduction, specifically Fourier's law, and adapted it to particle concentration. Fick's laws of diffusion basically proved that the rate of movement is directly proportional to the concentration gradient.
So he turned a qualitative observation into a rigid equation.
Yes, an equation we actually still use today in pharmacology to predict drug absorption.
The leap from a dead animal bladder to strict mathematical equations is just huge.
Yeah.
But the story gets even stranger when you look at osmotic pressure. In 1877, a botanist named Wilhelm Pfeffer builds these artificial semi-permeable membranes.
Yes, using clay pots.
Clay pots. And copper ferrocyanide, right?
Yeah, he essentially grew a membrane inside the pores of the clay.
Wow. So he meticulously measures the exact physical pressure required to stop water from moving into a sugar solution. He compiles all this amazing data, but from what our sources say, nobody really knew what to do with his numbers for a while.
No, they just sat there for about a decade. Then, a physical chemist named Jacobus van Hoff looks at Pfeffer's data and spots something extraordinary. The numbers perfectly mirror the ideal gas law.
Wait, like the equation you use for calculating the pressure of a gas inside a sealed container?
Exactly. PV equals nRT. Or, in the case of osmosis, pi equals mRT, where pi represents osmotic pressure. Van Hoff realized that dissolved solute particles in a liquid act, essentially identical to a gas confined in a room.
That is insane.
They generate pressure through their collisions against the membrane.
So dissolving salt in a glass of water behaves mathematically like pumping air into a tire. That is such a wild connection to make across totally different states of matter.
It was a profound realization. It actually earned Van Hoff the very first Nobel Prize in Chemistry in 1901. So, by the dawn of the 20th century, science had the math perfected. We knew exactly how to predict the pressure generated by these statistical collisions.
But the sources highlight a massive blind spot here. The math was perfect, sure, but for the vast majority of the 20th century, we had the actual biological mechanism of how water crosses a living cell membrane completely wrong.
Oh, yeah. We were wandering in the dark for decades on that one. The accepted paradigm was Ernest Overton's lipid solubility theory from 1899.
Right.
Overton argued that molecules cross a cell membrane based on how fat-soluble they are. The more fat-soluble, the faster they cross the lipid bilayer.
The glaring problem being that water is polar. It's not fat-soluble at all.
Oh.
And oil famously don't mix.
Exactly. So under Overton's model, water just awkwardly, slowly squeezes its way through the lipid molecules via simple diffusion.
It's like assuming a massive crowd of people is slowly oozing through the microscopic cracks between the bricks and the solid wall. It just doesn't make sense, especially when you look at certain human cells, like red blood cells and kidney cells let water through way faster than that squeezing through the bricks model could ever account for.
Yeah, physiologists knew there was a paradox. They suspected there had to be some sort of pore, but nobody could find it. It wasn't until 1992 that the mystery was accidentally solved by Peter Agree.
I love this story because he wasn't even looking for a water channel, was he?
Not at all. Agree's lab was studying Rh blood group proteins. As they were trying to isolate these specific blood proteins, they kept getting this mystery contaminant.
Like a glitch in the experiment.
Right. A 28 kilodalton membrane protein that just kept showing up on their gels. For a while, they just thought it was garbage, maybe a piece of a degraded protein.
But he eventually investigates the garbage and realizes it's the holy grail of cellular biology.
He discovers it is a dedicated water channel, which he names an aquaporin. It proved there was a hidden, high-speed tunnel through that brick wall the whole time.
Wow.
And structurally, it is a marvel of evolutionary engineering. The aquaporin channel has an hourglass-shaped pore. It narrows down to fit exactly one water molecule at a time in single file.
And it has an electrostatic filter inside it, right?
Right.
To stop everything else from getting through.
Yes, it uses specific amino acids to create a positive charge in the center of the channel. It lets water flip its way through, but absolutely repels positively charged ions. It is so precise that it won't even let a single proton sneak through.
Which is vital, because if protons could leak through, the electrical charge of our cells would just collapse.
Exactly.
And the speed of this passive tunnel is staggering. The sources estimate a single aquaporin allows 3 billion water molecules to pass through it per second.
3 billion per second. And again, without braining a single molecule of ATP, it fundamentally reclassified osmosis in living cells. We realized it sits on a spectrum, utilizing facilitated diffusion through these channels.
The physiological impact is immense, especially when you look at the human kidneys. I mean, every single day, your kidneys filter 150 to 180 liters of fluid out of your blood.
Which is an insane volume.
Right. But obviously, we don't pee out 150 liters a day. You dry out in an hour. We only excrete about 1.5 liters.
Because your kidneys rely on aquaporins, specifically regulated by an antidiuretic hormone called ADH, to passively reabsorb almost all of that filtered water back into your bloodstream. Agree actually won the 2003 Nobel Prize for the discovery, and it explains how we manage our internal water balance so flawlessly.
Which brings us to the real stakes of all this for you and me. Because these high-speed 3 billion molecule per second tunnels exist in our cells, the balance of solutes on either side of the membrane, a concept called tonicity, becomes incredibly powerful.
Oh, absolutely.
Get tonicity right, and it saves lives. Get it wrong, and it is rapidly fatal.
Tonicity dictates so much of modern medicine. Let's look at human red blood cells. If we take those cells and place them in distilled water, the distilled water is hypertonic. It has almost 0 solutes compared to the inside of the cell.
Okay, so the water outside is at a higher concentration than the water inside.
Right, so the water relentlessly collides with the membrane, finds those aquaporins, and just rushes into the cell.
It rushes in to try and dilute the inside until it reaches equilibrium. But a red blood cell doesn't have the volume for that, does it?
No, it doesn't. It swells up, the membrane stretches to its limit, and it literally bursts. That process is called hemolysis.
And doing the reverse is just as catastrophic.
Yeah, if you put those same cells into a heavily salted hypertonic solution, the water inside the cell rushes out to try and dilute the salty environment. The red blood cell instantly dehydrates, shriveling up into this spiky, non-functional shape. This is called crenation.
Which I assume is why a hospital will never hook you up to an IV drip of pure tap water.
Never. They use 0.9% normal saline. It is meticulously calibrated to approximately 308 milliosmoles per liter.
And that matches the blood.
Exactly. That concentration matches the tonicity of your blood plasma. If a nurse hung the wrong bag and gave a patient a severely hypotonic or hypertonic IV fluid, the osmotic shift could destroy their red blood cells or cause devastating neurological swelling in the brain.
This clears up such a massive misconception about hydration. We tend to think that drinking any liquid is hydrating, or that hydration is just about wetness. But as the math shows, wetness doesn't actually matter. The tonicity matters.
Right.
And the most famous tragic example of this is the shipwreck effect.
The historical terror of being stranded at sea. You were on a life raft, dying of thirst, surrounded by millions of gallons of water.
But seawater is incredibly hypertonic. It sits around 1,000 milliosmoles per liter, which is way higher than our blood. So if you drink it, it goes into your digestive tract, and instead of absorbing into your body, it acts like a sponge.
It osmotically draws water out of your body's cells, across the intestinal lining, and into your gut to try and dilute that massive salt load. You eventually excrete it, meaning you literally accelerate your own dehydration by drinking it.
It's a fatal misunderstanding of how our internal plumbing works. But our sources also highlight how flipping that exact same dynamic is responsible for one of the greatest medical triumphs of the 20th century.
We are talking about oral rehydration therapy, specifically in the fight against cholera.
Right.
Cholera is a bacterial infection that essentially hijacks the cells lining your intestines. The bacteria force those cells to constantly pump massive amounts of chloride ions out into your gut.
And because we know the rules of osmosis, we know water always follows the solutes, so the water chases the chloride into the digestive tract.
And the result is catastrophic diarrheal fluid loss. A patient can lose exceeding a liter of fluid an hour. It can kill an adult through pure dehydration in mere hours.
So if a patient's intestinal cells are actively dumping fluid outward, how do you hydrate them? I mean, you can't just have them drink regular water. It'll just flush straight through without being absorbed.
That was the massive medical challenge for decades. But the brilliant hack, which was refined by researchers in the latter half of the 20th century, bypasses that specific chloride pathway entirely. Doctors utilized a completely different cellular transport door called the sodium-glucose cotransporter, or SGLT1.
Okay, so how does that one work?
If you give a patient a precisely calibrated drink containing both sodium and glucose, this specific transporter binds to both molecules and actively pulls them together from the gut into the intestinal cells. Suddenly, the inside of those cells is highly concentrated with new solutes.
Oh.
Creating a new gradient. Water follows the solutes, so the water gets pulled osmotically back out of the gut and into the body, completely bypassing the massive fluid loss happening in the other direction.
Exactly. This one osmotic trick, the oral rehydration solution, distributed by organizations like the WHO, has saved tens of millions of lives, just by understanding the simple physics of how water moves.
That's incredible. Now, we've spent a lot of time marveling at the incredible power of osmosis for moving water. But what about standard diffusion for moving everything else, like oxygen, carbon dioxide, nutrients? If diffusion is so passive and efficient, why can't humans just rely on it to move oxygen all around our bodies? Why did we evolve the need for a heart to pump blood at all?
Well, that brings us up against the hard physical limits of the universe, which were elegantly laid out by Albert Einstein in 1905. Wait, Einstein was studying biology.
He was studying Brownian motion, that random thermal bumping of particles we discussed earlier. Einstein showed mathematically that the time it takes for a particle to diffuse doesn't scale linearly with distance. It scales with the square of the distance.
Okay, what does the square of the distance mean for you and me in practical terms?
It means diffusion is incredibly fast over microscopic distances, but uselessly exponentially slow over macroscopic ones. Take your lungs, for example. When you breathe in, oxygen only has to diffuse across an alveolar membrane that is about half a micrometer thick to get into your capillaries.
Okay.
That happens in fractions of a second. But if you relied on diffusion alone to move an oxygen molecule from your lungs down to your toes, a distance of roughly 1 meter, Einstein's math dictates it would take years.
Years for one breath of oxygen to reach your feet. You would suffocate instantly.
Exactly. That physical limitation is exactly why complex multicellular organisms evolved circulatory systems. We had to build a bulk transport highway, the bloodstream, because diffusion is only effective for the last few micrometers of delivery. Blood does the long-haul freight. Diffusion acts as the local delivery driver, taking the package from the truck to the front porch.
That makes perfect sense. And we harness this local delivery mechanism in everyday technology all the time, too. Like, kidney dialysis machines work by flowing blood on one side of an artificial membrane and a clean dialysate fluid on the other. It allows metabolic waste, like urea, to passively diffuse out of the blood, down its gradient, without losing vital proteins.
We use it in food preservation, too. Curing meats or making high-sugar jams creates a hypertonic environment that osmotically pulls water out of any invading bacteria, killing them before they can spoil the food.
Even large-scale engineering relies on it. Reverse osmosis desalination plants apply massive mechanical pressure to seawater, forcing the water backward across a membrane against its natural osmotic gradient, creating drinking water for millions.
It is fundamental physics applied to human survival.
Which makes the misappropriated science we see out in the world so frustrating. Reading through this research, I can't help but laugh every time I walk through a pharmacy and see those detox foot pads. Have you seen these things?
Oh, the ones that claim to draw impurities out of your body while you sleep.
Yes. The box always says something like, harnesses the power of osmosis to pull toxins from your body.
Yeah.
It drives me crazy. It takes this incredibly strict, predictive, thermodynamic equation, Van 't Hoff's pi equals MRT, and strips it of all meaning just to sell a glorified tea bag for your foot.
It completely ignores the underlying mechanics. Real osmosis requires a selectively permeable membrane and a genuine measurable concentration gradient of a specific solute. Toxins is not a scientifically quantifiable solute.
Right.
Furthermore, the thick, keratinized layers of human skin on your feet are structurally designed to prevent bulk fluid extraction in the first place.
It's the ultimate example of a scientific term sounding smart, so someone slaps it on a box to make a quick buck. I mean, if the biology worked the way they claimed, taking a bath would be a lethal osmotic event.
It highlights why understanding the why and the how of science is so crucial, rather than just memorizing a vocabulary word in a high school biology class.
Totally. So let's synthesize the journey we've been on today. We started by looking at diffusion and osmosis as the passive, statistical inevitabilities driving life. Trillions of random molecular collisions driven by thermal energy, pushing substances down their gradients without burning a single molecule of ATP.
We saw how those forces dismantle the idea of a mystical vital force, how they rely on carefully regulated gradients, and how our understanding of water transport was completely revolutionized by the accidental discovery of aquaporins.
We learned that getting these gradients wrong can pop a cell like a balloon or dehydrate a shipwreck survivor, but hacking those same pathways can cure cholera. And finally, we saw that these invisible forces are bounded by strict physical limits that dictate our very anatomy, forcing us to evolve a pumping heart to bridge the gap where diffusion simply takes too long.
It's a lot to take in, but if I can leave you with one final, active debate in physiology, something for you to really mull over, it's the concept of solvent drag.
Solvent drag, we didn't cover that.
No, we didn't. Throughout this entire discussion, we've painted a picture where diffusion and osmosis are these neatly separated, elegant mechanisms. Water moves this way through an aquaporin, solutes move that way through a different channel, and everything obeys its own specific concentration gradient.
Right, the tidy textbook version of the rules.
But modern quantitative modeling, especially in the incredibly high-flow environment of the human kidneys, suggests reality is much messier. Solvent drag is the theory that when you have bulk water flowing rapidly through channels or between cells via osmosis, the physical force of that rushing water might actually be dragging small dissolved solutes along with it.
Wait, even if those solutes don't have a concentration gradient pulling them in that direction?
Exactly. The sheer velocity of the osmotic water flow physically sweeps them along for the ride, like a rushing river carrying pebbles downstream. It means the perfectly tidy, isolated definitions we just spent all this time learning are already getting blurry under real extreme biological conditions.
I love that. It circles right back to where we started. The illusion of the biological traffic cops perfectly directing every molecule.
The closer you look, the more complex the microscopic landscape becomes.
The rules of thermodynamics are absolute. But biology is beautifully messy. It isn't a rigid machine. It is a roaring river of collisions and gradients, constantly passively working to keep you in balance. And you don't even have to think about it.
A statistical inevitability keeping us all alive.
Something to think about the next time you take a sip of water. Keep exploring, keep questioning, and we'll catch you on the next deep dive.